to put us all. So sorry about that. Anyway, it's a great pleasure to be part of this again today. What a wonderful day yesterday. Thanks to all the participants for coming and making this such a great, in a sense, community activity. So we had a series of wonderful keynote talks yesterday, and we're just delighted to extend them to this morning to welcome Dr. Aaron Gittler, whom we all know. Aaron is a professor of genetics at Stanford University. Many of us have had the pleasure of observing his extraordinary career trajectory since he was at the Whitehead Institute here in Boston. Several years ago, he has had a huge impact on how we understand neurodegeneration broadly, but certainly Parkinson's disease and ALS. He has published more than 150 publications in absolutely first-rate journals attesting to the quality and depth of his insight, and we're so grateful to have him with us today to talk about new insights into ALS splicing targets. Thank you so much for joining us, Aaron.
Thank you very much, Dr. Brown. It's really a great honor to be here to share with you some of the work from my laboratory. I changed the title around a little bit. I want to tell a story about a new TDP-43 splicing target, and then I want to tell a new unpublished story about 2 new ALS drug targets and perhaps 1 drug. And I'm looking forward to getting your feedback about that work. So today I'm going to talk about TDP-43, and this is the major disease protein in ALS. And I think this slide illustrates the problem. There are 2 sections of human spinal cord shown here. On the left, you can see the normal situation. This is a large motor neuron. This is the nucleus. I stained these sections with an antibody against TDP-43, and you can see here TDP-43 is localized to the nucleus, and here's the nucleolus, and TDP-43 is diffusely localized, and you can sort of imagine that TDP-43 as an RNA binding protein is binding to RNA, regulating splicing, other aspects of RNA metabolism. Then on the right is what happens in disease, and now you have 2 things going on. One, you have these large aggregates of TDP-43 building up here in the cytoplasm, either these round, dense, Lewy body-like aggregates or these ropey, skein-like aggregates. You can also see maybe these littler inclusions of TDP-43 out in the cytoplasm. So perhaps TDP-43 is causing problems by causing some sort of toxic cytoplasmic gain of function through aggregation in this out here. But then you can also notice that if you look at the nucleus, now TDP-43 is no longer there. So TDP-43 is not where it's supposed to be, so it can't do what it's normally supposed to do. So there could be a loss of function. So we should consider both a loss of TDP-43 function as well as a gain of toxic function in the cytoplasm. These 2 hypotheses are not mutually exclusive. They both could be contributing, but we should just think about these as we think about the drivers of disease. And I want to tell you 2 stories today, one about some insights into the normal function and the consequences of loss of TDP-43 from the nucleus, and then some insights into what happens when TDP-43 aggregates out in the cytoplasm and what we might be able to do about it. So as soon as TDP-43 was discovered as the major disease protein in ALS by doctors John Trochanowski and Virginia Lee from the University of Pennsylvania in 2006, and it was appreciated that it was an RNA binding protein, the first question that everyone asked was, okay, what RNAs does it bind to? So in a series of experiments from Don Cleveland and Jean Yeo, together with Magda Polymenidou and Clotilde Legier-Turin, they performed genome-wide analysis of all of the RNA targets that TDP-43 binds to throughout the mouse genome, and they identified thousands of these. At the same time, Rna Uli's laboratory did the same type of analysis, but in human neuronal cells and in human brain, and identified many targets for TDP-43. And in a breakthrough paper in 2015, Dr. Phil Wong from Johns Hopkins University discovered that a major function of TDP-43 is to act as a repressor of so-called cryptic exons. These are sequences in introns that are normally kept out of the mature messenger RNA, and TDP-43 functions to do that. It sits down on these yellow UG-rich motifs and represses the inclusion of these cryptic exons. And when you lose TDP-43 from the nucleus, it can no longer repress these cryptic exons, and now they creep into the mature messenger RNAs, and they can cause problems, you imagine, for several reasons. They might disrupt the reading frame. They could cause the RNA to become unstable and become degraded. they could introduce new peptide sequences that could cause trouble. Importantly, these are non-conserved. So if you do this experiment in mouse and you do it in human, you get cryptic exons, but they're different ones. So the specific ones are different. So what are the ones that TDP-43 regulates in human? So very recently, simultaneously, Don Cleveland and Clotilde Legier-Turin at the same time as Kevin Egan discovered that a major target of cryptic splicing target of TDP-43 is Stathmin-2, which you all heard about yesterday. So I'm not going to go into details of that, but other than to say, inspired by their work, we wondered, could there be other cryptic splicing targets out there that could be important for disease? And this study was led by a brilliant graduate student at Stanford, Rosa Ma. And what Rosa did, this was all during COVID lockdown where we couldn't go into the lab. We could just stay home and read papers and analyze data. And that's just what she did. She read this paper. It's a very important paper from Eddie Lee's laboratory at University of Pennsylvania. And what he had done was he took human brain from FTD ALS patients, and he stained them with the antibody to TDP-43, and then dissociated neurons, and then isolated the nucleus from those neurons, and stained them with the antibody to TDP-43, and then used this FACT sorting machine to separate the nuclei based on whether they had TDP-43 in there or not. And then he isolated RNA, and he did RNA sequencing, and he asked, what are the changes in genes that are expressed in nuclei that contain or lack TDP-43? And he found a host of very interesting gene expression changes. But Rosa didn't care about gene expression changes. She thought she could reanalyze Eddie's data, but look at it in a different way, look for novel cryptic splicing events. And she used two new alternative splicing prediction algorithms, and she identified 66 alternative splicing events that were occurring in the nucleus of neurons from human brain that lacked TDP-43. She found Stathmin-2 again, so that was good. She found HOLDIP-3, which had already been validated as a key splicing target of TDP-43. But then she found 64 of these other ones. The one that immediately caught her attention was UNC13A, because UNC13A is the top GWAS hit for ALS. If you don't include the chromosome 9p21 locus, which we know is C9orf72, UNC13A is the top of the list. This is the one that comes out in ALS. It comes out in FTD ALS. In a meta-analysis, again, if you don't include C9orf72, UNC13A is the one that rises to the top. In every review article about the genetics of ALS, we always hear about UNC13A, but we never knew how SNPs in UNC13A could contribute to risk for disease. But I think Rosa's figured it out. I want to show you the evidence. So first of all, here's the RNA sequencing analysis that she did when she reanalyzed those samples that she downloaded from the web from Eddie Lee's paper. Here is UNC13A. This is exon 20, exon 21. These are RNA-seq reads. So you see no RNA-seq reads, of course, in the intron, but in the nuclei lacking TDP-43, she sees RNA-seq reads, and she calls these CE for cryptic exon. She then went back to another data set she found. This is the supplemental data from one of the ClipSeq papers, and she found indeed there are ClipSeq peaks right in the intron, right next to the cryptic exon, suggesting that TDP-43 can bind here and repress that cryptic exon event. This is a hypothesis. She needs to do experiments to test that hypothesis. So she used shRNA to knock down TDP-43, first in neuroblastoma cell line, and then uses quantitative RT-PCR to detect the presence of the cryptic exon. So knock down TDP-43, cryptic exon goes up. She never detects it normally. And then the full-length UNC13A transcript goes down. And then by immunoblot, she can see that UNC13A protein is eliminated. This is neuroblastoma cells. She also does this in induced motor neurons from human iPS cells. Knock down TDP-43, cryptic exons go up. Full-length transcript goes down. Western blot, the protein's eliminated. So UNC13A belongs to a family of UNC proteins originally discovered in the worm C. elegans, because the mutants in those genes have this uncoordinated phenotype where because they have defects in neurotransmitter release. So this is, and the mouse gene is called MUNC13-1. Mice with mutations in MUNC13-1 are born but die shortly thereafter and are paralyzed, again, because they have deficits in synaptic transmission. So this is a critical neuronal protein that you don't want to lose. And now it appears that in cells with TDP-43 dysfunction, UNC13A protein levels are decreased because they have this cryptic exon that's disrupting the messenger RNA. She then extended her analysis beyond those 7 samples she downloaded from the web, and she collaborated with Len Petrocelli's team at the Mayo Clinic, and she analyzed every brain sample from the Mayo Clinic Brain Bank. These are ones with FTLD TDP. Everyone with TDP-43 pathology, she can robustly detect the cryptic exons. She analyzed a third data set. This is from the New York Genome Center. Here, again, she finds the cryptic exons in their RNA-seq data in the right brain regions. Importantly here, I think it's where she doesn't find it. She never sees it in cases with tau pathology or FTLD FUS, and never in ALS cases with SOD1 mutations, and of course, never in the controls. So it suggests that these cryptic exons are not just being formed as a consequence of degeneration, but specific to TDP-43 pathology. So RNA-seq reads and QRT-PCR is nice, but Rosa wanted a way of actually visualizing the cryptic exon in single neurons in the brain of patients. So she designed this single molecule fluorescence in situ hybridization approach using BaseScope, in which she designed a probe that flanks the canonical exon 20 and then the cryptic exon, CE. And she stained human FTD ALS brains and also stained them with the antibody to TDP-43. So you can see here, this nucleus contains TDP-43, it's normal. So she does not detect the cryptic exon. But 1, 2, 3, 4, these nuclei, you can see they have TDP-43 pathology. And then in 1, 2, 3, 4, they have the cryptic exons. Again, it's not subtle. Every neuron she's looked at, every nucleus that has TDP-43 pathology contains the cryptic exons, and then none of the controls that has normal TDP-43 ever has these. So it's a very robust marker of TDP-43 pathology, the accumulation of these cryptic exons. As a negative control, she makes another probe just flanking the exon 20, exon 21 to detect the full-length transcript. You can see normal TDP-43, lots of UNC13A messenger RNA. These 2 neurons that have TDP-43 pathology, you can get a sense that there's a less UNC13A messenger RNA consistent with her quantitative RT-PCR results. Remember I said it was the top GWAS hit for ALS. So we went back and looked at the locus, and this was pretty crazy. It turns out that the lead GWAS SNP is right in the cryptic exon itself. So UNC13A is an enormous gene. The SNPs could have been anywhere, but they're right here, right in this cryptic exon. So this immediately suggested to us a hypothesis how this SNP could increase risk for disease, perhaps by making this cryptic exon more prone to be included upon TDP-43 dysfunction. So Rosa was very clever. She realized that some of her samples that she downloaded from the web could be from individuals that were heterozygous for the risk in the reference allele. And if so, she might see an allele imbalance in the cryptic exon. And indeed, in our RNA-seq samples, she sees more RNA-seq reads with the cryptic exon harboring the G or risk allele. And then she did this in a systematic way with the Mayo Clinic Brain Bank data. And she could see as you go from 0 to 1 to 2 risk alleles, you have an increase in the cryptic exons. And then this is pretty crazy too. So then she does a survival curve. This is a survival of human patients, just stratified based on this this risk haplotype. And you can see this is FTLD, TDP. You can see the survival if you have 0 risk alleles, 1, or then 2 risk alleles. We're not the first ones to notice this. This has been known previously that this SNP can decrease survival. But what I think this says, I don't want to overinterpret these data, but what I think this says is, because we now know that these SNPs increase risk for disease by making this exon more prone to be included. I think it means if we can come up with a way of blocking the single cryptic exon event, we should be able to have at least this much survival benefit, but perhaps more, because these individuals, as you recall, also have cryptic exons in their brain when they have TDP-43 pathology. So we've provided evidence that UNC13A cryptic exons are a new facet of TDP-43 pathology in ALS and FTD. We wonder if there could be a way of defining cryptic exon profiles, STAFN2, which you heard about yesterday, now UNC13A, and perhaps some of the other ones. These might play out across the brain in different regions, and different patterns of these cryptic exons might underlie some of the different heterogeneity in phenotypes. So it'll be exciting to test all of these perhaps simultaneously in some sort of spatial transcriptomics approach. Are there genetic variations lurking in the genome that protect or promote cryptic exon inclusion in the face of TP43 dysfunction. UNC13A is the first one, but could there be other ones out there? And then, of course, therapeutic strategies. We know exactly where this cryptic exon is. We know where the splice acceptor site is. Could one design a antisense oligonucleotide to occlude that cryptic exon event? So even in the face of TP43 dysfunction, could you block this event? We recently published these data simultaneously with work from Michael Ward and Pietro Fratta, who also discovered this simultaneously. We find it notable that many other cryptic splicing targets of TP43 are involved in the synapse and in synaptic transmission. We have a team of postdocs tackling some of these other targets. It's a very exciting time in the lab. Hopefully, some of you can join us. There are many exciting projects that have been born out of this. So now I want to talk about the other aspect of TP43 pathology. We're just talking about what happens when you lose TP43 within a nucleus, but of course it's aggregating in the cytoplasm. And when I first started my laboratory, I used very simple model systems, budding yeast model system, in which we could easily recapitulate TP43 aggregation and toxicity and learn about the protein. But the power here is that we can then do genome-wide screens looking for suppressors and enhancers, modifiers of toxicity. And we've been doing that and finding genes that can allow yeast cells to grow better or worse in the face of TP43. And one of the best hits that we found is a yeast gene that has a human homolog, a related gene in human called ataxin-2. And ataxin-2, not only does it modify the phenotype of TP43 aggregation and toxicity, but it's already been implicated in another neurodegenerative disease called spinocerebellar ataxia 2. And the protein that the ataxin-2 gene encodes is a polyglutamine disease gene, which means like Huntington. There's a stretch of these amino acids, glutamine. And normally the length of the polyQ is 22, sometimes 23, greater than 34. It causes spinocerebellar ataxia 2. We hypothesized that perhaps something in between, longer than normal, but not past this range for SCA2, could be associated with ALS. So to test that hypothesis, we collected 1,000 ALS patient genomic DNAs, 1,000 healthy controls, and measured the repeat length. And we found that out of 980 control samples, we only found 4 individuals with repeats greater than or equal to 27. But we found nearly 5% of ALS cases having these intermediate length repeats. So we proposed that these intermediate length repeats in ataxin-2 increase risk for ALS. It's been exciting to see over the years that other laboratories around the world have been validating and replicating these findings in independent ALS patient populations. More work, we've shown that those intermediate repeats in ataxin-2, what they do is they make the protein more stable. So there's more of it around. So thinking about that, more ataxin-2 around increases risk for ALS. In experiments in model systems, like in yeast or together with Dr. Nancy Bonini and fly, if you lower ataxin-2, you protect against TP43. So could ataxin-2 be a good therapeutic target for ALS? So to test that hypothesis, you need to first perform experiments in mouse. So these are all experiments performed by a graduate student in the lab, Lindsay Becker, who's now a postdoctoral fellow at Princeton University with Dr. Cliff Brangwyn. And what Lindsay did was she crossed a transgenic mouse that expresses TP43 throughout the nervous system. This model is a very aggressive, fast progressing model of degeneration. The mice all died by day 30. And she crossed these to 2 independent lines of ataxin-2 knockout mice. And then she just analyzed wild type heterozygous or homozygous ataxin-2 knockouts. She found that the mice that were heterozygous for ataxin-2 had a significant extension in survival compared to the wild type mice expressing the TP43 transgene. But she found this dramatic extension in survival in mice that completely lacked ataxin-2. This was also sufficient to eliminate the pathology in the spinal cord. These are phosphorylated TP43 inclusions that are eliminated in the ataxin-2 knockouts. Here's the quantification. We really can't find any of them. We then collaborated with Ionis Pharmaceuticals to test a antisense oligonucleotide approach in which we can design an antisense oligonucleotide or an ASO against the ataxin-2 messenger RNA, deliver it into the nervous system of a mouse on day 1, and then see if this can rescue the TP43 phenotype. A single administration at day 1 was sufficient to lower ataxin-2 RNA and protein levels, and then it had this dramatic extension in survival with some of the mice living over 150 days. This video is anecdotal, but it shows you how powerful the rescue can be. These are 3 of those TDP43 transgenic mice at day 20. We injected all 3 of them on day 1. 2 of them we injected with a negative control, like a scrambled sequence that doesn't target anything. Those mice are paralyzed. This one we injected with the ataxin-2 targeting ASO, and this mouse is unimpaired and goes on and lives much longer. So we're very excited about how powerful this ASO approach can be. This is what keeps us up at night. TDP43 really is the major pathology in virtually all ALS cases, as well as almost half of the FTD cases. So we hope that targeting a modifier gene, ataxin-2, could be a powerful therapeutic strategy. And it's exciting that Ionis, together with Biogen, has launched a phase 1 clinical trial to test this ASO, or one designed against the human gene, in human ALS. We're not working with Biogen on that, but we're on the sidelines cheering them on. And we're very inspired by ASOs and their prospects in the clinic, which we can discuss at this meeting. But we also know that sometimes ASOs and gene therapy approaches have hurdles in delivery, in cost. And we're just thinking, could there be another way? Could we find a small molecule that can do the same thing, that we can just take, like orally available small molecule, to lower ataxin-2 levels? So I want to tell you 2 quick, completely unpublished stories about our progress to that end. They're by Garam Kim, a graduate student in the lab, and Caitlin Rodriguez, a former postdoctoral fellow. First, Caitlin's screen. What Caitlin did was she wanted to find regulators of ataxin-2. So before we can get to small molecules, we have to figure out how ataxin-2 is normally regulated. So she used CRISPR to insert this high bit tag in frame with the ataxin-2 gene in a human cell line. This is an inert 11 amino acid peptide, but then when you add large bit, it complements, it reconstitutes the luciferase, and you can detect levels using a plate reader. It's very sensitive. You knock down ataxin-2, you see less luciferase. She then used this in a genome-wide arrayed siRNA screen, and she found many hits. The top hit is the gene called RTN4R, which encodes the no-go receptor. You can see here, knockdown ataxin-2, there's less ataxin-2, knockdown RTN4R, just as much knockdown of ataxin-2, which is exciting. Here's a Western blot confirming it. So Caitlin thinks that this effect is at the level of the protein, because knocking down RTN4R has no effect on ataxin-2 RNA levels, and treating the cells with a proteasome inhibitor, MG132, abolishes the ability of RTN4R knockdown to decrease ataxin-2 levels. RTN4R, or the no-go receptor, is a receptor. There's a peptide inhibitor, NEP1-40. Indeed, when we add these to cells, it very rapidly lowers ataxin-2 levels. We then did experiments in mouse neurons or human neurons. These are primary mouse neurons or iPS-derived neurons. Treating with shRNA to knock down RTN4R lowers ataxin-2 levels in mouse as well as human neurons, shown here. Same thing with the peptide, adding increasing concentrations of this peptide in mouse neurons, decrease ataxin-2 levels. Same with the human iPS neurons. And then Caitlin analyzed RTN4R knockout mice, and you can see heterozygous and homozygous knockouts have less ataxin-2 in their brain. So this is just what we're looking for. This knockout is viable and fertile. They just have less ataxin-2 in their nervous system. So perhaps RTN4R could be a therapeutic target. Garam Kim is a graduate student in the lab. She also wanted to find regulators of ataxin-2, but she wanted to do it in a different way. She designed a FACS-based, fluorescence activated cell sorting based CRISPR screen. No tags, no nothing. Just takes human cells that have Cas9, transduces them with a genome-wide CRISPR library, makes all knockouts, fixes them, stains them with the antibody to ataxin-2, sorts them into ones that have high or low levels of ataxin-2, gets the DNA out of them, sequences the barcodes, finds which knockouts had them have less or more ataxin-2. You can see here that in some of her validations, the screen worked. She finds knockouts that lead to less cells having as little ataxin-2 as when she knocks out ataxin-2 itself. But then she gets every single subunit of this lysosomal vacuolar ATPase. Whenever you do a screen and you get every subunit of a complex, it tells you that complex is important for whatever biology you're studying. You can see here, knocking out one of the subunits, the cells have less ataxin-2. This is specific. Knocking out this ATPase doesn't affect levels of other polyglutamine disease proteins like Huntington or ataxin-1. It seems specific to ataxin-2. The other reason this caught our attention is that it's already the target of a FDA-approved drug that's been used to treat osteoporosis. It's called atidronate. And this has been approved by the FDA for over 40 years, and it's been there's decades of safety and real-world data on it. So we got some of this atidronate, and we first treated mouse neurons. And you can see here, again, it's very safe. We can treat a wide range of concentrations. They just have less ataxin-2. In mouse neurons, this is just 3 concentrations, but really a wide range of concentrations. There's less ataxin-2. Same for the human iPS-derived neurons. It's not toxic, and they have less ataxin-2. Then we thought, why don't we just feed it to mice? So we just did this experiment where we put in the water of the mice, or this metagel, this gel-like food they can eat. And then this is just a 1-week treatment, and we see that they have less ataxin-2 in their brain. So it's safe, they can tolerate it, they just have less ataxin-2 in their brain. What's exciting is atidronate is a bisphosphonate. There are many other versions of the bisphosphonates in this class. We've been testing all of these. And there's another one, which you may have heard of, alendronate, which is marketed by Merck as Fosamax. And this one also is able to lower levels of ataxin-2. Right now, we're testing this compound in a ALS mouse model, and we'll report the results as soon as we get them. So we hope the ASOs work, but these are going to cost hundreds of thousands of dollars per year, and gene therapy approaches might cost millions of dollars. We got 1 gram of atidronate for $50. So we hope that these other approaches can make treatments for ALS more accessible. We're also very excited about the prospect of using genome-wide screens for regulators of levels of ALS genes. TBK1 is one where you might not want to lower it, you might want to raise it. So Olivia Gauthier did the same screen that Garam did, but now looking for things that increase TBK1 levels, and she finds knockout of this phosphatase leads to increased levels of TBK1. So we're very excited about this type of levels-based screens. I'll stop there, acknowledge great collaborators along the way. The ataxin-2 story, discovering the mutations and the modifier of TB3 pathology, was a joint collaboration with Nancy Bonini's laboratory. Study with UNC13A is a joint collaboration with Len Petrocelli's lab. I've collaborated along the way. Happy to answer questions, and looking forward to further discussions.
Thank you very much. That's another extraordinary line of work and talk from you, Aaron. We're very, very grateful. Let me back up and ask a little bit more about the UNC13 story, if I may. Can you say a few words about, and you alluded to it briefly, the physiology of UNC13, the neurotransmitter release, and how that relates to survival in ALS?
That's a great question, and one that we're thinking a lot about because the mice, at least, or when you knock down UNC13A, it doesn't cause degeneration. So, but it does cause defects in synaptic transmission. So, but actually, there's accompanying our paper and the Frada paper, there's a News and Views article from Noah Lipstein, a world expert on monk proteins, and she has an interesting hypothesis where she said something like, imagine you have two neurons innervating muscle, and say one of them degenerates, but the other one is just fine. It can communicate, synaptic transmission communicate, but now that one has that one has TP53 pathology, and so it can't undergo synaptic transmission. So that may mimic what would happen as a degeneration, but it would just because it can't communicate with other neurons or muscle. So in that way, perhaps deficits in synaptic transmission can cause signaling defects, functional defects.
Interesting, very interesting. Thank you. So there's a couple of questions in the chat. Nitesh Sangai from Manitoba asked, what's the rationale of using HEK293 cells in the initial screening that you did in the second part of your talk? Did you think of using spinal cord motor neuronal cells such as NSC34s?
Yeah, that's a great point. And this is always where we have to strike a fine balance. On one hand, the 293 cells are very plentiful, and they divide quickly, and they're easy to handle, and we can do screens very rapidly. On the other hand, they're not there might not be as disease relevant as the spinal cord motor neuron cells that you propose. So it's sort of a trade-off. My approach has been to do screens with very simple systems first, get a bunch of hits, then dive deeply into those hits in those other perhaps more relevant systems, rather than building the screen with maybe the perfect system up front. It's just a different approach.
Thank you. So two other questions regarding, again, this rather extraordinary a second part of your talk. One is, can you describe the mechanism of action of the bifostinates in reducing the lysosomal ATPase?
That's right. That's a good question. The structure of the bisphosphonates, they kind of mimic ADP in a way, and I think they may act as a competitive inhibitor for ATP hydrolysis. I'm not yet an expert on bisphosphonates, although I'm trying to learn more about it.
Thank you. And I had just a very, very simple question. What can you tell us about the brain, the CNS permeation of, for example, alendronate or atidronate?
Yeah, again, I don't know. These compounds were selected because they go mostly to bone. So the reason I showed those other series is we want to find maybe alendronate or atidronate are the ones that will work, but maybe there'll be other ones that can be delivered in a different way or might be able to get to the brain. But the other thing that I think we should also consider is that there are cell non-autonomous effects. There are systemic effects that could lead in an indirect way to downregulation of ataxin-2 in the nervous system. So these compounds perhaps need not get directly into the brain. But to answer the question directly, we don't yet know how much of this is getting into the brain.
Yeah, it's very interesting. Just as an aside comment, which is it's many people, including several on this call, have done screens, for example, in mice of attempted repurposing of all FDA-approved drugs. And it's not so clear that has been as fruitful as we'd like. But when you have a highly specific, focused assay of the type you've developed, then the screening, which is CRISPR-based, becomes so much more precise and so much more informative. It's very interesting, the precision versus broad screening, single molecule versus whole animal, a hugely different set of results.
I would love to do this screen in human. So for example, if this drug has been around for 40 years and many people have been taking it, if there's a way of comparing electronic medical records to see if this offers any sort of protection, against motor neuron disease, that would be an exciting area, I think. But then it might also just be worth finding trying this type of thing or designing a clinical trial to test this.
Yeah, very, very interesting. Aaron, just mind-blowing work. Thank you so very much. This is just fantastic.
Wow.
Great way.
Thanks for having me.
That's great. So we'll move on now. We're just delighted that the rest of this day will be a series of presentations from our colleagues in the world of biopharma. This is yet another high point for this symposium. We'll start with a presentation from Exaltis Pharma, from Sigel Lemarchand, if I've pronounced that correctly, the head of preclinical R&D. The title is Multi-Properties Peptide NX210C for the treatment of ALS. Janet, do we have Exaltis here?
No, sorry. Can you hear me?
Great. Yes.
Oh, good.
Sorry, the presentation was planned in 20 minutes. So I will share my screen. So hello, I am Dr. Sylvie Lemarchand. I'm head of the preclinical department at